All right. That sounds like a good plan.
So, well, it's a plan.
That sounds like a plan.
There we go. That's all I needed.
From ScienceSortOf.com, you're listening to Science Sort of.
Hello and welcome to Science Sort of. This is episode 299. Our theme this week is Phone a Friend, and I'm your host, Ryan Haupt, and joining me to discuss things that are science, things that are sort of science, and things that wish they weren't science. And joining me to talk about that is Sarah McAnulty.
Hey, how's it going?
It's going well. So Sarah is a Ph.D. candidate at the University of Connecticut, studying how immune cells in squid interact with and recognize their beneficial bacteria, amongst other things, and is the founder and developer of Skype a Scientist. So, Sarah, we've been trying to get you on for a while, and I'm really excited to get a chance to talk to you.
Yeah, I'm super happy you had me on. Thanks.
So we're on Skype, but do we want to talk about Skype first, or do we want to talk about your research first?
It's up to you. We can do either.
Okay, well, since the show is named Science Sort of, we tend to start with the science and then transition to the sort of.
Sounds
so
great.
Let's talk about some science. I am a vertebrate paleontologist, so I eschew your research organism in the very name of the science that I do, but I will tell you that I love squids, and I'm very excited to talk to you about squid.
Love talking about squid. Can do it all day.
Okay.
That's great.
You say on your website, which we'll link to all this stuff in the show notes for the episode at scienceworld.com, but you say on your website that you study host microbe interactions, but what kind of scientist do you describe yourself as when asked?
When asked, I usually say I'm a squid biologist, but the part of the squid that I'm actually studying is how their immune system interacts with bacteria.
Okay, so you're a squid immunologist, squid microbiologist?
I would certainly say I'm a squid immunologist, but I think typically when I'm talking to just a family member or a member of the public, I usually say squid biologist.
Awesome. And you had a video that was produced by some other podcast that I'd never heard of called Science Friday.
Yeah.
That was a joke.
I know.
So you have this great little video. It's like a 5-minute video on YouTube that I will link to as well in the show notes if people want to see the little squiddies that you're working with. But can you tell us, you seem to work primarily with a squid called the bobtail squid?
Yes, I work with Hawaiian bobtail squids. So these guys are really cool because they have a bioluminescent species of bacteria, so they like glow, that live in a specialized organ on the underside of the squid that's appropriately called the light organ. So when the squid first hatch, they need to recruit that bacteria from the seawater. And so what I study is basically how this partnership between the bacteria and the squid maintains itself in a healthy situation throughout the life of the squid. And so the reason that they keep this bioluminescent bacteria in their bodies is because they're nocturnal. And so they want to use the light from the bacteria to effectively hide at night against light coming down from below. So they have all of these predators swimming beneath them. And these predators are looking up, and they're looking for like silhouettes that are above them so that they can attack the silhouette. And maybe it's going to be a fish-shaped silhouette or a squid-shaped silhouette. And the bacteria, that light, breaks up that silhouette so that the predators can't see the squid.
So is that sort of how like great whites think that surfers silhouetted against the sun or moon look like seals?
Exactly, yeah. So if we just smeared ourselves with really bright bacteria, maybe great white sharks wouldn't eat us as much.
I think there's a market for bioluminescent surfboards here.
Absolutely,
That's
yeah.
a great idea. We should delete this recording immediately and start developing this new business model.
Immediately make a patent for that, yeah.
So, we've never had a squid biologist on before. So can we talk a little bit about squids more generally and give people a little bit of just like squid background?
Sure. So squid are members of the cephalopods. And so cephalopods are octopus, cuttlefish, squid, and nautilus. Nautilus are kind of the odd guys out here. And so squid are invertebrates, so they don't have any backbone. They first evolved and showed up in the fossil record 500 million years ago. And so that's longer than trees existed. That's before sharks. It's when all life was in the ocean.
Is that cephalopods or squid specifically?
That's a good question. That is cephalopods broadly. So that's when the first shelled cephalopod came up off the seafloor.
And cephalopods are like a type of mollusk. They're related to like oysters and snails and stuff.
Yeah, exactly. So the squids specifically are mostly pelagic, which is to say that they swim up in the water column. But the bobtail squid that I work on, they are just sort of couch potatoes. They like to sit on the seafloor and bury in the sand all day long. And so there are like over 300 different types of squid, and they're all really different from each other. There are some that are massive. They're like 40 feet long and really, really, really heavy. And those are called colossal squid, and those live in the Southern Ocean. And then the giant squid, of course, is skinnier than the colossal squid, but a little bit longer. And then there are itsy bitsy tiny little squid that live in the Indo-Pacific called pygmy squid. And these guys are like less than an inch. They're like 1.5 centimeters long. They're tiny. So basically anywhere in the ocean you can think of, there's probably a cephalopod there. And oftentimes there's a squid there. So they're really cool.
So they sound like as diverse as mammals almost.
Honestly, yeah, almost. I mean, they're all aquatic, but other than that, they're...
Well, let's hope they stay that way. I don't.
Yeah, exactly.
If squid start evolving to live on land, we are screwed.
We're officially in trouble. Yeah.
And squid they're closely related to things like octopus, but what makes a squid and not like an octopus or a cuttlefish?
Sure. So octopuses are off on their own branch. They diverged quite a while ago. And so octopuses have 8 arms, no tentacles, and their body plan is just a little bit different. So their mantle, so that's the part of their body where all of their organs are, is shaped differently than it is in the squid and the cuttlefish. And so squid and cuttlefish are more similar to each other than either is to an octopus. And they have a little bit more structure to their body. So for cuttlefish, they have what's called a cuttle bone. And that's effectively like an internalized shell. And that shell is made of aragonite, and it's full of air, and that helps the cuttlefish stay buoyant in the water. So it doesn't need to put a lot of energy into moving up in the water column or going down toward the seafloor to stay exactly where it is. For anyone who might be a scuba diver, if you're familiar with your BC, that's that like thing that you put on to stay neutrally buoyant.
Buoyancy
they basically
control
have
device, yeah.
Exactly, yeah, they have one built into their body, and so that's also reasonably hard in their body, and so that gives them structure.
Some of the more advanced BCDs, the buoyancy control devices, have integrated weight systems too, so you don't have to wear the weight belt. So that's more
awesome.
Like the cuttlefish.
Yeah, exactly. So cuttlefish are super cool. And then there's squid. It's like hard to say one thing about what a squid is because they're all quite different from each other. But for the most part, they're kind of torpedo shaped. Some of them have this internal structure called a pen, which is also your like structure giving internalized shell, but it's a lot smaller and more flexible than the cuttlebone is. And then the squid that I work on is sometimes called the dumpling squid. And that's because they legitimately kind of look like a dumpling. They're just sort of round. They have absolutely no structure inside them.
Really? So they have no pin, they have no pin bone or anything?
No pen at all. Yeah.
Wow,
all they have that's hard is a beak. Everything else is squishy. So that makes them.
So they're pretty derived amongst squiddies.
Oh, totally. Yeah.
And I guess I was surprised to learn that no other squids have this sort of specific bioluminescent coevolution. Right.
So there are a lot of bioluminescent squid, but most squid make that bioluminescence themselves. So there's this one squid called the firefly squid that makes its own light. And it has light at the ends of its tentacles, and then also all on the underside of the body. And some of the photophores, they're called, those light-producing organs, some are blue and some are green. And they're just like so gorgeous. And so those live off Japan and are just like a dream. And then there's some squid that also live deeper, that also have at the end of their tentacles, mostly to entice prey. And then the squid that I work with use bacteria to make the light instead of making it themselves. But there are other animals that have this symbiosis with bioluminescent bacteria. So if you're familiar with that lure on the anglerfish, the light from that lure is also coming from bacteria.
Oh, really?
Yeah.
Oh, that's cool.
Yeah.
So our squids, or maybe cephalopods. Cephalopod means head foot. I've never quite known how they got that name.
It's because, when you think of a vertebrate or most animals that you'll see at the zoo, it's kind of like head, shoulders, knees, and toes. And if you look at a squid, that is not the case. You've got arms or tentacles or both, then eyeballs, then torso. Like it's totally differently arranged. And so the reason they're called head foot is because their eyes and where their face sort of is directly attached to their arms or their feet. So instead of us having our eyes and feet on the complete opposite sides of our bodies. They're right next to each other in the cephalopods.
We have a frequent guest of the show who's an echinoderm person, so I might get in trouble for asking this question, but are cephalopods the best invertebrate?
I mean, you're asking a cephalopod person, so I certainly think so. I think they're awesome.
I'm tempted to agree with you, but I won't go on the record for the purposes of unit cohesion over here in the studio. Yeah. And then so maybe this is a little bit morbid and I shouldn't ask it, but do you eat squid? Like, do you have calamari when you go out?
So I will occasionally eat dory toothus, which is like market squid, but I don't eat cuttlefish and I don't eat octopus because I've raised those two animals from eggs. And once you raise a baby animal to an adult animal, I don't know, I can't kill them after that. But that's just me.
They're also stupid smart.
They are stupid smart. Yeah. So the market squid, I've had them in the lab for a brief period of time. And while they seem smart, it's not the same kind of personable smart that I've seen in a cuttlefish or octopus. So they're like harder to connect with kind of. And so I think that might also have something to do with it.
So is the market squid named because it's the squid we typically eat?
That is, yeah, exactly.
Wow. Awesome.
It's also called like the long fin inshore squid. So that's like more about its body and where it is. These common names, like every animal can have more than one common name, but the market squid is what we normally call the group of squid that we eat.
Excellent. Okay, so getting back to the specifics of your research now that we've covered some squid basics, it's really cool in the video, so I hope people will go check that out. But basically, the bobtail squid has this sort of glowing ET heart inside of them that helps them, like you said, breaks up their silhouette. And you also said it mimics the color of the moon?
Sort of. So the light from the bacteria is one color, and then it goes through filters to let only a certain color of light out. So it mimics the brightness of the moon more than the wavelength of light. So the squid can tell how bright it is or dark it is outside using photoreceptors on their back, and then can control the amount of light that comes out of where the bacteria live out to the environment, which is super cool and very impressive.
And your PhD work is focusing on, because these squids, when they hatch, they're not hatching with this bacteria already inside them. So they somehow have to encounter the bacteria, and then the bacteria has to get into their little glow sac successfully without being destroyed by the squid's immune system, and then subsist there happily for the lifetime of that squid. So that's a pretty complex interaction. One of our co-hosts is a parasitologist, and she loves to talk about the complexity of parasitic interactions, but this isn't even parasitic. This is everybody's working together, but they're having to work around some pretty significant hurdles, right?
Yeah, exactly. So when the bacteria and the squid first encounter each other, the bacteria basically has to go through like a gauntlet to get into the squid, because there's currents that are moving right around where they have to enter that only certain sized particles can really get through because of these cilia that are beating on these like two appendages, these like extensions off of the light organ that are only there during the recruiting phase of the squid's life. And then after that, they get into this mucus area that's full of toxic stuff to most bacteria. Well, to the gram-positive bacteria, there are two kind of big, broad brushstrokes distinctions of bacteria. One is gram-positive and one is gram-negative. And the glowing bacteria are gram-negative, and the gram-positives can't survive in this mucus. So, okay, that cuts out a good chunk of the bacteria that may live there. And so after that, they have to go swim actively down these ducts, and they're getting hit with acids, and they're getting hit with other chemicals like nitric oxide. And eventually they get down into the crypt where they'll survive for the rest of their life. And that's when they've officially made it. So it's not as though any old bacteria can get into that light organ space.
And how do they encounter these bacteria once they've hatched?
The correct bacteria is about 1 in every 1,000 cells. So the squid just sort of swim around. They're constantly breathing in seawater because squids' bodies are open to the seawater in what's called their mantle. So that's like their main body part. And so basically it's just a matter of filtering. And that's so cool. Eventually 1 or 2 of the right bacterial strain are going to get in there and then proliferate.
So it's not a crazy uncommon bacteria. They just have to swim around and they'll encounter it. Because 1 in
they'll
1,000 when
eventually
it comes to
bump into
bacteria is probably...
That's a lot.
Yeah.
Yeah. It's not that common in the whole ocean, but in the areas where these squid live, there's more of it because every dawn, the adult squid will eject about 95% of their bacterial population in the light organ. And we think that seeds the population for coming generations.
That's awesome. That's really cool. And then like squids, I mean, they're basically little jet engines, right? They're like sucking in water and then spitting it back out their siphon, and that's how they power around the ocean.
Exactly, that jet propulsion.
Which is why they are torpedo-shaped, because they function like a little biological torpedo.
Yep, exactly.
And now we know that when they explode, they don't explode with destructive force. They explode with bioluminescent wonderfulness.
Absolutely.
And so it's interesting because it sounds like you've had to become as much of a microbiologist as like an invertebrate biologist.
Yeah, absolutely. I have. It's been a learning curve.
And then you're studying how the immune system, which is the cells within the squid that are meant to keep bad bacteria out, how they are not accidentally attacking the good bacteria that are trying to get to the glow sac.
Exactly. And I've been working on trying to understand what it is about a symbiont, so the bacteria's cell, like how is the immune cell identifying that bacteria. And so I've been looking at the structures on the surface of the bacteria and other things that the bacteria can do and what sort of informs the hemocyte, the immune cell, as to what to do.
So is the hemocyte like our white blood cell?
Exactly, yeah.
And then when you say looking at those white blood cells recognize pathogens by like the surface proteins on the outside of the cell. And so you're trying to figure out how they know to give the good bacteria, wave them through into the club, basically.
Exactly, yeah.
And so how do you actually figure that out? Like, what's your experimental procedure?
We will take blood out of the squid. So we'll bleed them non-lethally. So we just knock them out, take like a tiny, tiny drop of blood out of them.
Well, now this is a show that I don't mean to cut you off, but this is a show that has a segment called What Are We Drinking? So I'm going to have to ask you to specify how you knock them out.
Okay, no, sure. That's a great question because we knock them out with alcohol. So we put ethanol in their seawater, so the like small transfer tank that we move them in. And then when they've been in that water for a little while with the ethanol, they'll just sort of turn pale. So the color changing cells on squid and octopus and cuttlefish are called chromatophores. And basically they're like tiny sacs of pigment. And when they're active, they can sort of pull the sac into a pancake, and then you can see the color that it is. And when it's contracted, it looks just like the tiniest little pinprick. And so when the squid is knocked out, those big pancakes turn into tiny little pinpricks, and the squid looks like almost see-through. And that's when you know that the squid's knocked out. So once we do that, we can pull them out of the water very, very briefly, use a needle to pull out, for any scientist in the audience, like 20 microliters of blood, which is like, if you picture an eyedropper drop, like a third of an eyedropper drop. It's very, very...
These squids are not huge too, right?
They're like lime-sized. So if you take a teeny drop out of a lime, the lime's fine. And so
every time you describe these squids, they get cuter. Bobtail, dumplings, little limes.
They're like rainbow colored too. They're like literally the cutest things you've ever seen.
So I think way back in the early days of this show, so this is episode 299, so we've been doing it for a little while, we had somebody suggest a tagline for the show called Making Science Drunk Again. And I think this is the first time where it is the thing that is being studied that is being made drunk and not the scientists we're talking to.
Yep.
So that's
very accurate.
Pretty hilarious to me.
Yeah.
Because the ethanol, like, it doesn't, I mean, obviously you couldn't do this if it was hurting the animals, so it stuns them, but it's not having like a long-term negative effect on their life.
Exactly. They bounce back within minutes after you put them back into the seawater without ethanol.
So they don't even have hangovers.
Well, I've never asked them, but I hope they don't have hangovers. I think they do okay.
I mean, in the video you show them the sort of when they're in their pale mode, and then you kind of show them coming back to life when you reintroduce the water. It's very cute because you like squirt fresh water over their gills, sort of like to wake them up a little bit, like the smelling salts in the the movies.
Legit, yeah.
And then they really do just kind of brighten up, like whoop, and they turn. It's like the squid turns back on.
It does, yeah.
Because do squids sleep?
So cuttlefish sleep. So that leads me to believe that yes, squid sleep. I don't know. So like the squid that sit on the seafloor, I think the answer is a pretty resounding yes. But I don't know how animals that are swimming constantly sleep. I know in whales they like turn off half their brain,
Sure.
and then that half sleeps, and then the other one switches. But like a mammal brain and a squid brain are quite different, so I don't really know how the heck that works. But I know there's been study on cuttlefish.
But weirdly, a squid eye and a mammal eye are actually quite similar.
Oh, I know. It's so weird and cool. Convergent evolution, man.
Yeah, it's almost as if
Amazing.
Evolution is a thing that really
I
happens.
know. Yeah.
Not to turn this into a
An
dialogue
Evo,
on.
yeah.
Yeah, exactly. But so you've done this kind of crazy convoluted study where you're taking the blood, and then what are you looking for once you get the blood?
Then we take out the blood cells, those hemocytes. And so that's super easy because they're the only type of blood cell in the blood. And then from there, I add this.
Because they're invertebrates. They don't have like red blood cells with hemoglobin, right?
Right. So the blood is blue because of this oxygen binding protein, hemocyanin, which is copper based instead of iron based, like our blood, which makes the blood blue. And so it's really easy to pull our squid immune cells out of there because it's just this free floating protein, as opposed to like a human where we have red blood cells and that oxygen binding protein is like trapped inside of a cell. So that makes them also really easy to work with from an immunologist's kind of point of view. So once we get the cells out of the blood, we then stain them with this fluorescent dye called cell tracker. So it's a dye that will get sucked into cells and then get trapped in there and turn fluorescent. So it's a way for me to be able to look at the immune cells under a microscope without having to like do any genetics to make them glow, which is just super convenient. And so from there, I can put them on a microscope. I use a confocal microscope. And then I do time-lapse videos with different types of bacteria in with those immune cells. And so I can, for example, knock out a certain protein or something on a behavior, for example, on the bacteria, and then watch how that changes how the immune cell treats that bacteria. And see, for example, if it will destroy fewer of those bacteria or something like that.
That's awesome.
Yeah.
And so what have you been finding? Or you don't have to talk about results if you're not ready to talk about results.
Yeah, I'm not allowed to talk about results,
okay,
unfortunately.
fair enough.
But
No, that's exciting. That means eventually there can be a sequel to this conversation, I hope.
There will be, yeah.
Okay. So instead of talking about the results that you're finding, but we'll just assume for the sake of argument you're finding really cool stuff.
Cool stuff is happening, I promise, yeah.
We will take your word for that for sure. So instead, I want to wrap up our sciencey part of the talk with a more general question of what's something you wish that people knew about squids or understood about squids that doesn't seem to be in the public awareness?
I think one thing that is probably not good for the squid for me to tell everybody this, but generally speaking, squid are really good things that we should be eating because squid populations refresh every year or even more frequently than every year. And we've been overfishing the oceans really badly. And so fish can take a really long time to regenerate and for their populations to rebound. And squid grow every year. Every squid has like 1,000 babies, if not more. So they're a more sustainable fishery than fish fisheries. So get used to squid because we're going to be eating more of them probably.
I've heard Alton Brown say the exact same thing.
Yes, I can picture the Alton Brown episode you're talking about where he's got like, yeah.
Yeah. So both food professionals and scientists agree. Eat all the squid you can.
Eat squid, yeah.
Now, do you prefer that with a spicy marinara or more of the Thai chili sauce? Is the real question.
I prefer it as ceviche, so put a little lime on there.
Yeah.
It's really good, yeah.
I'm a big fan of ceviche. Apparently, it's a great way to get parasites, but
oh, bummer.
I know, right?
I hope that doesn't happen to me.
This is the problem when you have a parasitologist as a friend. You learn about all the avenues by which parasites get into your body. So apparently acid, great for cooking seafood without heat, not great at killing their parasitic load.
Noted.
But speaking of parasitic loads, we're going to take a quick break to cut over to a drinking segment and then come back to you to talk about your outreach project.
Sounds good.
Black and white, all of the contours are laid out before me now to leave a trace. with all of the light and shade. We take up our own space. I'll find my own way back to the past tense. A lifeline to highs and lows, to seeing the bright side, and I should know. We wait for the afterglow to cover the blind side, and I should know.
Okay, well, it will not be an episode of Science Sort of if we did not also talk about what we are drinking. So this is Ryan, and we're going to take a break from my interview with Sarah, and joining me on this siesta, I guess, can you be awake if it's a siesta? It's Abe. Abe's here.
No.
Has to be asleep to be a
Hi,
siesta.
everybody. You must be asleep and not doing work in order for it to be a siesta.
Okay, so we're going to take a siesta now.
Peace.
I mean, but I guess first we should have a drink before we take our siesta.
Oh, that would help. Yes.
Okay, Abe, would you like to go first, or would you like me to go first?
Sure. I'm happy to go first.
Okay. We shared the same drink last night,
We did.
which is rare for us, but I allowed it.
It's true, but it was a limited list of cocktails, and I didn't feel like ordering off the menu, and what you picked sounded excellent. So a rare confluence of all of those things.
Yeah.
So today, before this siesta, I'm actually going back to, I guess, our most traditional drinks, which is a beer. And I have a seasonal beer that I've been hanging on to. I'm not sure why, because this would have been just perfect in the summer, but I decided I'm going to drink it now. And it's actually really good. So I'm drinking a seasonal beer from Blue Mountain Brewery. They are Nelson County, Virginia. And so it's called Native Species, and it's a hoppy Belgo-American ale.
Well, that's a good choice, because we were talking about squids, which are native to the oceans. So they're a native species. I guess they're not a species. Squids are an order.
But they are native.
But squids are a group. They are a native. True.
It's a native group, yeah. So
Yeah.
I figured we could somehow relate this back to talking about native things.
Totally.
To native people.
Well, my beer is also hopefully thematic with the discussion that Sarah and I were having, because we were talking about the bioluminescent bacteria getting inside the squid. And so I have a beer called the Inside Job IPA from Henhouse Brewing in Santa Rosa, California, which was a brewery that Julie and I visited 2 summers ago, but I wasn't able to bring anything back because we were just on like a quick little day trip, road trip thing. So we just stopped in to visit the brewery and really enjoyed their beer. The brewery had been recommended to us by some locals. But when I was back in California for the holidays, Jesse came over and brought some beer and then forgot the beer and then left. So now I have leftover beer from Jesse's overambitious beer bringing tendencies.
Well, that is very fortunate. Helps to have friends like that, right?
It sure does. And this is it's just a very, I've been lamenting for months on the show now the overabundance of East Coast style IPAs in my life. And so to have here a crisp, cold West Coast IPA in my hand just makes me very happy. Yeah.
I debated between having an East Coast IPA and decided with going the local route, and just I've had a few beers in my fridge that I've been saving for recording days. This was one of them. And
funny how that happens.
Right. It's kind of, it sort of dictates my decision making at the beer store, at the bottle shop. But I've been trying to go local support the last few months whenever I've picking up beer. I'm still new to the Virginia and general like greater DC region, mid-Atlantic coast breweries. And so it's been a good way to explore. It's like, all right, which of you sort of local or at least regional breweries make a beer with a name that's great for recording?
Awesome. Well, we are recording this now over Skype, and in our next segment, Sarah and I are going to talk about a program she started to get scientists on Skype called Skype a Scientist.
Yay.
So that's coming up next.
Tonight in a bar alone I'm sitting, apart from the laughter and the cheer. Scenes from the past rise before me, watching the bubbles in my beer. Visions of someone who loved me brings a lone silent tear into my eye. And I know that my life's been a failure, watching the bubbles in my beer.
So in addition to being a squid superstar, you are also the founder of Skype a Scientist. Your name is Sarah, you study squids, and you do things on Skype. You have a lot of S's in your world.
I can't believe I've never noticed that, but that is true.
So tell us about Skype a Scientist, because that's how you and I actually first kind of got to know each other and what sort of prompted me to ask you to come on the show in the first place. And then when I found out that you were a squiddy, I was also super excited. I hope squiddy's not an offensive term to anybody, but
I don't think so. If it is, I haven't heard of it yet.
Okay.
But yeah, so I started this program. It was right after Donald Trump got elected.
Who? I'm not familiar.
This Cheeto that runs our company. Well, so he's treating it like a company, our country. So yeah. It was
did he inspire people to take activism to the next level in their own personal everyday lives? Interesting.
I think he did. He sure did for me. So yeah, I noticed that there was just this like dread and anxiety in the scientific community about what could happen to the country, what could happen to people, what could happen to science. So I felt like there was all of this energy that needed to be funneled towards something good. And so one thing I knew that scientists, as like a broad brushstroke community, needed to get better at was talking to people, because I figured, you see people doing juice cleanses, you see people advocating for not vaccinating your kids. There's like a lot of pseudoscience that's been going around in the last, say, like 10 years. And so I felt partially like, okay, we know we need to increase and improve science communication, and we know that everybody's all kind of revved up right now. So how can I kind of take advantage of this moment? And so I started Skype a Scientist because I figured that schools would be a kind of easy inroad to start with getting people involved. And I asked teachers if they wanted to get paired up with a scientist, and scientists if they'd be down to talk to students. And then from there, it just totally ballooned from there. We had about 800 classrooms our first semester and 500 scientists. And now we get about 5,000 classrooms a semester. Yeah. And our scientist numbers vary because when I hit a certain number of teachers, I just go on Twitter and say, yo, I need more scientists, and a bunch more sign up. But the problem that we currently have, which is a good problem to have, is that scientists are so enthusiastic about this program that they keep signing up again and telling their friends. And it's getting out to the scientists faster than it's getting out to the teachers. I think part of it is that I'm a scientist. I constantly talk to scientists. I'm constantly talking about the program. And so it just sort of like ripple effects out. So we can accommodate about twice the number of classrooms that we currently have signing up. So about 6 months ago, we started fundraising so that I could basically put out Facebook ads for teachers to hear about the program. And that's been hugely successful in upping our teacher numbers. It's not that the teachers don't love it. Our feedback from teachers has been overwhelmingly positive. And yeah, they just love having a view into what a real scientist's life is like. A lot of times scientists will kind of carry the computer around their lab and do like a little virtual field trip into what a real scientist's lab looks like. And teachers have really responded well to that.
Oh, that's cool. I haven't thought of doing that yet. That's a good idea.
Yeah, it's fun. It's fun. One thing that the program really wants to accomplish is just having everyone that encounters the program be able to say, like, I met a scientist. The thing about this program is that instead of having the scientists deliver information to people, it's a conversation, because we don't want to just like have scientists and science as like an establishment, just be this like fountain hose of information going to people. We want to hear what people have to say, and we want to hear what people care about. So if you're having a back and forth conversation in a Q&A session, you can hear what your students actually want to know and then answer those questions and have them feel like they're being heard and not just like talked to, And so that's one benefit of the program.
I love it. Because of doing this podcast for the number of years we have a number of teachers who are listeners, and I think they even sometimes play clips for their classrooms. And so some of them have reached out to me personally before I started doing Skype with Scientists and said like, hey, will you Skype with my classroom? And I always say, yeah, sure, I'd love to. And those have been great experiences. So when I started, I don't even know how I first saw this opportunity. I maybe we were already following each other on Twitter, or maybe you posted some listserv or something like that. But I somehow, the Skype word in whatever context of like science Twitter or my emails caught my eye, and I was like, oh, this is a thing that I've been doing personally, but like I'm not that organized about it, and this seems like a much better system to get it done in a way that actually like benefits hopefully the teachers and the students' schedules even better, and I get to reach out to classrooms that I would never have access to before. And so it's been a really positive experience for me. So I've done it 2 years now. And my first year, I had a little bit of trouble because I don't think I understood the demands on teachers' time as much as I do now, given some other experiences in my life. But the second year I did it, I had a couple of really good classroom chats, and it is so much fun. And something I think about is the number of times in my life where I'm out in the world and people ask what I do, and I tell them I'm a paleontologist, and they say usually one of two things. They say like, oh, like Ross from Friends, which I usually hang my head in because, as we all know, Ross is a monster. And but they'll other times say, oh, I've never met a paleontologist before. And it has happened often enough that it's hit me that like, oh, people really don't have any sort of daily or even lifetime level of interaction with scientists.
It's totally true. Yeah. I mean, I think a lot of people, when they picture a scientist, they just picture what they see in the movies. And so we're either completely socially awkward, like we're the professor from Back to the Future, or we're the guys from the Big Bang Theory, totally socially inept, or like we're dorks, or just straight up evil. And that's just not the scientist population I know. And on top of that, usually in the movies, we're white men. And when we're not white men, we're white women. Like, how often do you see a scientist in a movie that's not white? It's extremely rare. If you just Google scientist, it's all a bunch of white people, usually in a white coat looking at like a swirling blue liquid or whatever. And we know that that's just not true. And so we want people to see scientists as they truly are, and of all like the various diversity that we have in the scientific community. And so
are you yourself a participant?
I've participated every semester, even though I haven't signed up every semester because so we allow teachers to specifically request scientists, and they can just toss a name in there. And if they haven't signed up, sometimes I'll just like try to find the person and ask them if they want to participate. And so I've been requested through that. And I've Skyped at least one classroom every semester so far.
And so what are kind of a two-part question, like what are the diversity of types of, like not just, I hope you're getting good gender and racial diversity in your scientists that are signing up, but like what are the disciplines of science that are being represented in the scientist side of thing? And then like what geographic and age range of classrooms are they being able to reach?
We've got diversity basically no matter how you slice it. So we have scientists so in the beginning, it was pretty biologist heavy, and now we're really branching out and getting a lot further. So we have physicists, we have like meteorologists, chemists, we've got social scientists, we've got scientists who only speak sign language. We have scientists that only speak Spanish, French, whatever. We've got languages other than English for sure. We're in Europe, the US, South America, Central America, Africa, Asia, we're pretty much everywhere. We have more countries represented in the classroom side, but we still have like 20-something countries worth of scientists. So we have, I think, 65% of our participants on the scientist side are women. And then from there, it's like a huge breakdown. We've got like, it's just amazing to think that we could have a teacher say, I would like a Latin American lesbian, and I can be like, done. No problem. Got it. It's like really kind of amazing. So I think we've done a good job of reaching a lot of different pockets of science and not just stuck with ecologists, which is what we had at the beginning.
That's awesome.
Yeah.
Have there been any just crazy stories that have come out of the experiences people have had working with teachers and students? Like any scientists that have come back to you and said like, oh, the wildest thing happened to me and it was just totally not what I expected?
We've had a lot of cute things. Like one scientist came back and said like the class brought their bearded dragon up to the computer, their like little pet in the classroom, which was really cute. I know we had one teacher tell their scientist, who then told me that they had a student who like wasn't really engaged and wasn't really caring much, and then had the session and really saw themselves in the scientist, and now is like maybe thinking about going to college and wasn't really necessarily thinking about it before, and like majoring in science. And so that's the kind of story that we really like to hear. But also there are some cases where like, yeah, maybe it didn't change what the kid wants to do with their life, but it took them from being actively averse to science to being like, scientists are okay, and that's like, we'll take it. Like that's kind of amazing, because really we just want people to view science as not a scary, bad thing. So even if they don't want to become a scientist, if they can view science and scientists in a better light, then we've succeeded.
I've had that exact attitude towards students in actual classrooms that I've taught in, where I'm just like, I'm clearly not going to convert you. I'm not going to change your major after taking this, required oceanography course. But if I can get you out the other side of this course with a positive feeling towards science and the scientific method, I'll count that as a win.
That's a total win. Yeah.
Because, there's no jobs anyway. So like, go do something.
So true.
so go play in the NBA, which is what that student did.
Well, geez, I wouldn't turn that down. That sounds great.
That was my other line of thinking of, is like, this person is going to have influence over a lot more and a very different crowd of people than I'm ever going to have influence over. So like, let's make them positive towards science if we can.
Totally. That's super important.
Yeah.
Yeah.
What are the age ranges of classrooms? Because the ones I've done have all been on the younger side.
Yeah, so we have students as young as kindergarten. And then we actually go all the way up to adult. So we've had a group of friends I think they were in Boston. There was like 7 adults that got together and had a session with a scientist, and they had a lot of fun. We have a lot of high school classrooms, and then the scientists can also tell us what age ranges they're comfortable working with, because maybe somebody like really wants to talk to a 3rd grade classroom, so we can give them a younger group. And then others are like, I'm a theoretical physicist, and I just kind of want to talk to AP physics classes. So give me the seniors in high school.
I'm a theoretical physicist, nothing but 3rd graders.
Yeah, exactly. If they can talk to 3rd graders, I just think theoretical physicists, that sounds like the hardest job I can think of, but that's coming from an immunologist. And so maybe they think immunology is hard. Who knows?
Our in-house theoretical physicist is one of the best people at coming up with a convoluted but helpful metaphor to explain any physics topic you need. So
that's awesome.
The Titanium Physicist Podcast, for anyone out there needing a physics podcast.
Sweet.
And then the job that I imagine is the most daunting for you is you have these applications coming in on both classrooms and scientists. How do you end up doing the pairings?
Great question. So I have my best friend since like high school is named David Jenkins. He just got his Ph.D. from Boston University, and he wrote me a Python script that will match them all up. So every 2 weeks he runs the script, and then the script will spit out all of the matches from that round for everybody that's signed up in the last 2 weeks. And if there's any classrooms that don't get matched, those get spit out to me, and then I match them manually the best I can. And so that's really awesome. He's really made a really fantastic system for this. And so what I personally do is make sure people are signing up, make sure people who are having problems get those problems fixed, help with funding and marketing and yada, yada, yada. And he does the actual matching itself using computer science. And so
that sounds like a pretty amazing friend to have.
He is amazing. I literally don't know what I would do without him.
Unless there are any videos of him dancing gleefully while at Boston University, and then I don't want anything to do with him.
Oh my God. No, I don't think he busts a move on video. If only. Yeah. I actually went to Boston University as an undergrad, and when I saw that video, I was like, oh, hey, I know where she is. That's
a good
Marsh
video.
Plaza. I see in the background. Yeah.
Ridiculous.
Yeah.
We're not even going to explain what we're talking about. Everyone can just,
you either know or you don't know.
Because this episode's going to come out in 48 hours, and it won't be a news story anymore.
Right.
Because that's America right now. So it sounds like you actually have a pretty elegant system for matching everything up with, some effort put in on your end as well. Because I know we have both scientists and teachers who listen, can you give, I feel like we've made a pretty good pitch for why this is a beneficial thing to both groups, frankly, but what do people do next if they're really excited and they want to get involved?
If you want to get involved, it's pretty straightforward. You just go to skypeascientist.com. Right on the homepage, there are links to get signed up for either a scientist or a group leader, which would be like a teacher, or an adult group leader, a scout leader, or whatever. And then it'll send you to a Google form. You'll fill out all the information and then just hang tight until you get a match. We do matches once every 2 weeks. So you should get a match within 2 weeks.
Awesome.
If you're a teacher. Scientists may have to hold tight a little longer. It's kind of a luck of the draw type situation.
So like I said, I've done it twice now, and you clearly iterated it smartly in ways that it is getting better and better with each iteration that I've done it. So the form, the Google form you fill out, like it is not overly long, but it is comprehensive in a way that is really helpful, I think, for making sure that everybody gets the best experience out of it possible. And I see now that is this new to this iteration about the chatting with adults in correctional facilities?
That was new about halfway through last semester. We had one scientist that said it was really important to her that we include that. And I thought that was so awesome. And it hadn't even occurred to me. So we included that. And she said that she was going to reach out to correctional facilities to see if they would sign up. So it's in there, but I don't know if we've actually had any sign up yet, but we're absolutely open to that being a thing.
I just searched through my email and tried to find my original time filling out the form to see because there's a how did you hear about us, and I was trying to figure out how I heard about this thing, and I cannot find the emails. So but it's great because like you get to be pretty specific about the type of science you do. You get to explain the languages that you speak. You get to be very precise with your schedule. But I think my favorite thing about this form, and I wish this was a thing we could make scientists click the box for almost any time, like I try to submit a paper with them or something like that. And it's by clicking this button, I solemnly swear that I will either contact my match teacher or communicate that my teacher needs to be rematched. So like you're
taking like an oath.
You're really trying to beg scientists to like answer your email.
It's true. Yeah. But honestly, that's probably the biggest problem in the whole program. People not answering their emails.
I think that's one of the biggest problems in the endeavor of what we call science.
In human life. Yeah.
It is science,
right?
Just science. I think I've dealt with people who aren't scientists. They seem to be handling this email thing fine. I don't know what we did in our weird culture of being scientists that we're bad at email, but here we are.
Here we are. Yeah.
Okay, cool. So rolling enrollment. So anyone can sign up at any time, either teacher or scientist. You try to match them up every 2 weeks. What else do you want people to know about the program, or what else do you want to share with us, everyone listening?
The last thing, which is super important, is that we're currently raising money to run the program full time.
Yes. So sorry. I can't believe I'm so sorry. I forgot to mention that.
That's okay. We're mentioning it right now. No problem. So I am a Ph.D. student. I will be graduating with my Ph.D. in August of 2019. And we want to run this program full time because it is a full time job, without a doubt, running this program. And so I've been supported by my Ph.D. salary, and I won't be in 9 months. So we need to figure out a way to keep this machine rolling after I graduate. And so we do a number of things at Skype a Scientist other than just match classrooms. So we match about 5,000 classrooms every semester, totally free to the classroom. We also provide these Skype a Scientist live sessions. So they are sessions where we'll bring in scientists that we know are really awesome communicators, and we'll do YouTube live sessions with these scientists. And you can submit questions ahead of time, or you can submit them in the session. And we also do this new program we're running called Skype a Scientist After Hours. Those are just for adults that are contributors on our Patreon page. So we bring in a fun scientist that isn't afraid to curse or talk about mating behavior, for example, on the second Wednesday of every month. And I'm doing the first one. And then we've got frogs. We've got a person who studies and talks all about eating bugs, for like humans eating bugs, and why we should all be doing it more. She's one of our other speakers later in the year. We also organize live shows in Boston, New York, and Philadelphia. And I want to, after I graduate, do science communication training for scientists, like in little modules that are totally free to the scientists as well. There are some science communication training things out there, like the Alan Alda Institute, but those are all paid. Like you need to pay for them. And I really want to develop things that can be done from anywhere for people with really busy schedules to get them better at communicating without having to pay, because there's really not that much money available for making yourself better at science communication. And so I want to help scientists help themselves.
What I tell scientists is just when you're an undergrad, start a podcast, do it for 10 years. And then you'll be okay.
Yeah, no problem.
You'll be just half decent at it, and people will still give you mediocre iTunes reviews. Like, it's
there
fine.
You go.
It's good.
Just jump into it and try it. Yeah. So that's all that we do. Yeah, exactly. We do a lot of, I think, really important programming at Skype a Scientist, and I need more people to donate to our Patreon so I can keep it going. And like even $1 a month is wildly helpful if we get enough people to do it.
Well, that is at patreon.com/SkypeAScientist, all one word. And people can go there. We have a Patreon for our show as well. But for this episode, I will dedicate ourselves to pushing people to your Patreon instead, because I think what you're doing is really cool. I'm really excited to be a part of it. It's really frustrating me that I cannot remember how I first found out about this and how we first started communicating about you coming on the show. Mm-hmm. Because I found the email that I sent you being like, hey, I saw about this thing you do and you should come on the podcast. And that was like 2 years ago.
Oh, my God.
I know, right? So I'm glad we finally were able to get you on the show, because it's been a pleasure talking to you about everything you do. And you have a well-curated and well-developed internet presence. So you have a website.
Thank you. Yep.
Which is Sarah McAnulty? Sarah McAnulty.weebly.com. You also have a Twitter, Tumblr, Instagram, and YouTube. So your Twitter is Sarah McAttack, where you talk a lot about the Skype a Scientist and the things that it needs to move forward into the future. I haven't checked out your Tumblr or Instagram, but I assume they are delightfully squiddy.
Super squid all the time. Yep.
And then we will also link specifically to the Skype a Scientist subpage on your website, where people can sign up to be either a scientist participant or a classroom participant. And I think this is also one of those things where even the folks listening who might not be scientists or teachers, you probably know one. So let them know about this as a resource they can tap into. I have been working with a teacher in my local neighborhood, a high school science teacher, and I have found that while they are supremely capable of getting the job done, every little bit that the community can help them do the job is beneficial to both them and the students. You're taking a little bit off their plate, and you're enriching the education of the students that you're interacting with. I have had wonderful experiences doing Skype a Scientist. I often end the call wishing it could have gone twice as long, but it's like my attention span is a little bit longer than the students that I talk to in most cases. So I get that they don't necessarily need to hear me keep talking about sloths for another 20 minutes, but every experience
sloths
I've had,
are great. Yeah.
Sloths are great, but when they ask a good question, I have to answer, and that can take a minute. So
yeah,
and they often have very good questions. So I've really enjoyed my experience with it, and I thank you for taking a thing that I was doing haphazardly and systematizing it in a way that works for many, many more people beyond just myself. So kudos to you for all the hard work you're doing. And I hope that as this program develops and as your research continues, you will come back and tell us, give us updates in the future.
Sounds good. Thank you so much.
All right. Thanks for joining us.
No problem. Bye.
So that was my conversation with Sarah. Thanks again to her for her time talking to me about her research and outreach, which is a great 1-2 punch that we love to talk to scientists about here on the show. But when we talk about those things, you often talk back at us, and we feature that talking back in a segment we lovingly refer to as the PaleoPOW. PaleoPOW is the final segment of the show where we feed back the feedback and hopefully don't create a very harsh, sharp noise in the microphone. Hey, speaking of creating sharp, harsh noises in the microphone, Abe, you just gave a talk last night.
I did, yeah.
How'd that go?
I thought it went really well. I was only over by about a minute.
As opposed to the 20 minutes you were worried about being over.
15. Let's not exaggerate.
Yeah, so
Yes.
you gave a talk at the first meeting of 2019 of the Geological Society of Washington, an organization we have both become members of in the past year or so. And you did an excellent job. And then it actually, you set up the next speaker very nicely. And then we got to hang out with the next speaker, Joyce, a little bit afterwards. And she also lives in the DC area. And I think she would make a great guest for the show, since you two could come on and talk about your integrated, well, not integrated in an actual sense, but integrated conceptually research.
Yeah, so I spoke about some of the work that I've done in Iceland with mid-ocean rift and hotspot coupling of magmatism, and Joyce focuses on modeling mid-ocean rift upwelling of mantle magmas. So it was very appropriate, and it was a lot of fun.
And I realized that as much as we've had you on the show, we've never actually had you talk about your dissertation work. So sometime in the near, not too distant future, you'll have to come on and actually tell us about your dissertation, since that's clearly an oversight on our part, not having had you talk about that before.
Yeah, that's actually an interesting thought. We've talked about Iceland a lot.
Because I was sitting there in the talk and I was like, I haven't heard most of this before. It's weird that this is new information to me.
I mean, to be fair, the first time I was on the show to talk about Iceland, we talked about sort of an intro to the work I was doing at the time, which did not include some of the oxygen isotope in-depth analysis that I ended up doing towards the end of my PhD. So
the oxygen isotope part
right.
of the story was the thing that I had a question about, but then you got cut off because you took too long answering the other question, so I didn't get a chance to ask my question.
Oh.
So I'll save it. No,
do you want to ask it now?
I want to save it for the show. Even though we're doing the show right now, we're not doing that show right now.
Well, we'll save it for that show.
The show we're doing right now is the final segment of episode 299, Phone a Friend. And we have a friend in Tyler D, who has become a Patreon of our Here podcast. He went to patreon.com/science-sort-of and signed up at the Avogadro's Army level or higher. So we have to thank Tyler for, I assume, his support of the podcast.
Yeah, awesome. Tyler, thank you so much for sharing some of your hard-earned money with us.
And I don't know much about Tyler. Someone with Tyler's name followed me on Twitter a few years ago, and so I have how they describe themselves on Twitter, which was as a person who works in the pharmaceutical industry in the southwestern portions of Ohio, and they are a Christian guitar enthusiast and avid procrastinator. So, Tyler, if that's not you, tough. We're making some assumptions here. So, but we also have to tie it in to what's going on with the episode we just talked about. So, we have to do something with Squid and Skype. So I'm thinking...
And inside jobs.
Well, the inside job would be nice, but I'm thinking maybe we could look, or maybe Tyler, this is Tyler's thesis, was Tyler looking at maybe like the pharmacological effects of making Cincinnati-style chili out of squid?
Oh, yeah.
So how do we turn that into a thesis? Because one of the things we talked about in the interview, I know you haven't had a chance to listen to it yet, Abe, is one thing that we can do that will actually help ocean health is to eat as much squid as possible. Like, squid reproduce very, very quickly. It's basically impossible to overfish them.
Ooh.
So we should all be eating squid every chance we get. And as you know, I'm a big fan of Cincinnati-style chili, the greatest of the regional chili styles in these United States.
I see. I don't think that I've ever had it.
I don't think you ever have either. And so I think we need to do a Sky on Chili Day.
Yeah. Is there a good barbecue in Cincinnati?
Is there a good barbecue?
I mean, I kind of got spoiled in Nashville, but...
Yes, there's a place called Montgomery Inn, which does fantastic ribs.
Oh. How about Squidbecue?
Well, but I think...
Squilly.
But barbecue...
Squiddy chili.
I mean, we talk about barbecue, we talk about like the meat falling off the bone, but squids don't have any bones.
They have a beak.
Yeah, but I think you would want to remove that. I don't think
What if it was like squid falling off the beak?
There are cultures that do barbecued squid, I think in places like Korea, but I want to...
I've had it in Japan. It's actually really good.
I believe it's really good. But, I was just thinking like squid chili.
Okay, that's since it's the greatest chili
what about this?
in the United States.
All right, here's what Tyler does. Tyler cuts... Because Cincinnati-style chili, as everyone knows, is served on top of spaghetti. And so what if
I learned something new today.
What if the arms and tentacles are the noodles, and then the bodies are chopped up into the mincemeat to make the chili?
That sounds great. How do we put that into a scientifically sounding?
Well, because we're testing the pharmacological effects.
Squid spaghetti.
Okay. Oh, yep. I see where you're going there. Cephalopod spaghettification.
Yeah.
Cephalopod.
The effect.
Because spaghettification is a real thing that happens in black holes. But I think it also is fair if you're turning something that wasn't spaghetti into spaghetti, it should still count.
So the pharmacological effect of cephalopod spaghettification, a regional approach to ocean conservation.
In the preparation of Cincinnati style chili.
Squid chili. No, okay, you can do in the preparation style. That I think that covers
Cincinnati style chili.
It doesn't necessarily need to be traditional chili. Yeah.
Oodles of so pharmacological potential pharmacological effects. We don't know there's going to be any potential pharmacological effects of cephalopod spaghettification in the preparation of Cincinnati-style chili, do oodles of noodles decrease oodles of squids in the world's oceans?
I like what you did there. I think that's good. Bong.
All right. Tyler, potential pharmacological effects of cephalopod spaghettification in the preparation of Cincinnati-style chili. Do oodles of noodles decrease oodles of squids in the world's oceans? ?
Tyler, congratulations on your dissertation.
Making chili is a great thing for a procrastinator to do because you literally just put everything in a pot and wait. So if Tyler really is an avid procrastinator, right, he puts it on the pot and then, or even better in a slow cooker, because then it's set it and forget it, and then goes and plays some guitar.
Yeah, that's excellent. There's two things we have in common, Tyler. I like to play guitar to avoid doing the things that I should be doing.
And Tyler and I have in common, I hope, we presume that Tyler is as big a fan of Cincinnati-style chili as I am, but maybe not. Tyler, let us know.
Yeah. All right.
Abe, what do you got for us?
I have a hello note email. That was an email? Yeah. An email from our friend Gary K.
That's 1,000 Garys.
There is 1,000.
Killigary K. K is
the... Yes, the 1,000th Gary.
First in our heart, 1,000th in the Gary.
In the Gary world. Yes. First in ours, though. And Gary writes to us to share some thoughts about GSA 2018, which took place last November in Indianapolis, Indiana. He actually writes to Ryan and he says, "Hello, Ryan. I really enjoy listening to the show. Did you get a chance to go to GSA this year? I thought you would like my notes from GSA this year. This was my first conference, and I really enjoyed it. Have you ever made coffee with an AeroPress? I recently got one, and I'm anxious to try it out. Have a great weekend. Keep up the good work with the podcast. Gary K." So I did go to GSA actually after a multi-year hiatus of not going to conferences. My Ph.D. advisor retired this year, and he had an honorary session because he received a career award from GSA. So I got the opportunity to go back to GSA and give a talk in his honorary session. And I thought it was actually a really nice session, but also good venue. I'd never been to Indianapolis before. That was interesting.
I did not intentionally make this a Midwestern PaleoPOW, but I seem to have done it.
But it is now. Yeah. So that's awesome, Gary, that you got to go to your first conference. I think that was one of my favorite aspects of science outreach and sharing science with other scientists. It was kind of like the highlight of, here's what I'm doing, and here are people that not only know something about it, but care deeply about it. Not that they're the only ones, but it was always a great forum to come together with other minds that are thinking about the same processes and exchanging ideas. So that's awesome.
Yeah. And I mean, anyone's first meeting is a intense and overwhelming experience. My first meeting was actually also GSA many moons ago. I presented a poster, but I had basically no idea what I was doing there and didn't really know how to attend the meeting. And I certainly was not as productive as Gary, because Gary sent us some of his notes that he took, which is awesome.
And impressive. Yeah.
Yeah. A really productive way of engaging with the meeting. Not something I ever even attempt, because I'm usually usually when I'm preparing something for a conference, that's taking up my entire attention. And by the time I'm at the conference, I'll maybe get the evening before everything starts to look through the abstract program and figure out what talks or sessions I'd like to go check out. And I usually just find like a session that is generally, 70+% of the talks in that session are what I'm looking for. And then I'll just camp out there. And I will maybe jot notes in the program book that's given to us. My only like hack that I do at conferences to help me keep myself organized is if we get a badge that comes in a little plastic sleeve. If I get anyone's card during the meeting and I want to follow up with them, I put the card in the plastic sleeve of my badge holder. That way, when I get home, I have a direct link between like, oh, these are the cards I got at that meeting because that's the badge from that meeting.
Yeah.
I don't know. What do you do? Do you have any tips and tricks for first-time conference attendees?
I don't. I feel like the evolution of my attendance at these conferences has been so traumatic. Like, I remember the first conference I went, which was AGU many moons ago as well. Over a decade now. And it was just so overwhelming. And I think really when I think back on the stuff that I've learned over a decade of attending conferences, one of the things that sticks out in my mind is I felt the need to take up every minute of my time at a conference with talking science and conferencing. Mm-hmm. And the more I've attended meetings, I've realized it's a really overwhelming and mind-tiring task to try to do that. And so for my own sanity, I always schedule myself either a day off or some time to not think science and instead do something else that gets my mind to rest. And that usually involves having happy hour or drinks with people I know, which is great because there's a lot of people I know I don't get to see outside of these conferences. And, there's a lot of social events after hours. And so very often I'll take a day off to kind of go explore wherever the city happens to be, like Indianapolis, and just kind of roam around, maybe go see the exhibits. There's always fun stuff at the exhibits, and really try to give my mind a rest, because it can be incredibly tiring to, just have your brain 100% on the whole time you're there. And, I like to tell people it's okay to check out for a day.
I always tell myself I'm going to do that, and I'm terrible at actually doing it. And then I get burnt out every single time. I can't.
Yeah.
I'm bad at it. I'm bad at pulling myself away from the meeting, even if it would mean for a, on the whole, more meaningful meeting experience. I just don't do it. So it's good advice, but not advice that I'm myself capable of taking.
Well, and it's a hard recovery afterwards, right? It's like putting in double overtime for an entire week, or, in the case of something like GSA, 5 days instead of, well, I guess it's still three or 4 days instead of 5 or 6, plus travel if you have to travel, plus everything else. And it adds up very quickly.
It doesn't help that the most recent meeting we had was AGU, which was here in DC. And so it sort of felt like it was all hands on deck all week because I was just trying to, like, you and I were there technically covering the meeting as press. So we have a bunch of content that we recorded together that's coming out in the next couple of weeks. We're going to do, a special episode for 300. And then after 300, we're going to start with all of our AGU coverage. So you and I were recording interviews. We had events most evenings. There were multiple alumni events. We had a Vanderbilt alumni event. Bev and I were helping put together an AWG event along with a lot of other organizations. So it just, you can very easily get yourself overbooked in a pretty severe way, which happens. Yeah.
Well, and we had a long workshop that you and I, Ryan, hosted for science communication and outreach from the podcasting perspective, how to get started on a podcast. And even organizing things like that, where Ryan has done it in the past, I have not. That took up a lot of my effort and energy as well, just trying to think of, how can I be helpful in running this workshop.
And another thing I would say to someone like Gary is that the first time you go to a meeting is almost the most freeing because you don't have social obligations and you may not necessarily even have research obligations. And so you can kind of just absorb the meeting on your own terms. And as you continue to go to meetings over and over again, that iterative process of like every time you go to a meeting, you meet more people that are likely to be there the next time you go to that same meeting. And so every time you go, you're going to have more and more people that you want to see and touch base with and go see their talk or go see their poster. So it really kind of snowballs as you head into your scientific career, and not in a bad way. It changes the dynamic of what your goals are when you go to a meeting. especially a giant meeting like GSA.
It's true. And that also has been an incredible evolution in my own attendance, in that the first couple of times I went, I was interested in seeing talks. The last 2 times I went, I was interested in tracking very specific people and asking them questions about my research, not theirs. I wasn't really interested in what they were going to talk about or what they've been doing. But, there are people that are prominent enough in my field that I thought I could use their thoughts on my own work. And so things like that, you start to organize. And so GSA for me was twofold. One, go and see my advisor in this honorary session, receive his award and do his retirement. But also there were 2 or 3 people. That's really all the people I talked to, 2 or 3 people that I wanted to track down and say, hey, we've never met, but I work in this. You know my work. I know your work. Here's a question about my work that maybe you have some insight about. So it does become more productive towards your own work rather than just observational. And like Ryan said, that's not a negative thing. It's just there's a little bit more purpose the more you attend these and the more you realize what kind of connections you can make, what kind of information you can exchange and things like that.
Cool, man. Well, Gary, thanks for writing in. And in the future, most of these meetings have started to transition, especially the larger meetings, to having searchable abstracts that you can just put in a name. So if you're wondering if we're going to be at a meeting, just search and see if we're, I tend not to go to meetings at this point unless I'm presenting something. So odds are if I'm at that meeting, you can find my name in the abstract booklet. And so if you are able to find my name and can track me down at a poster or a talk, come say hi. We're always super excited to meet people who fans of the show when we're at these big meetings. It really does mean a lot.
Yeah, and same here. There was a second question. I have had a coffee made with an AeroPress, but I've never made it. And I got to say, for all that I remember about it, it was very smooth and very enjoyable.
Yeah, my mother-in-law had an AeroPress for a while, and that was what she was using to make coffee. But I never quite figured out how to use it myself, and she has a pour-over cone, so that was what I ended up using whenever I was there making coffee. But it seems like a cool system. It seems similar to the way they do the Vietnamese coffee when you go to a Vietnamese place. Yeah,
and I'm partial to the French press, which is what I use at home. So I've never had the need for...
And that was what you used when you were a coffee professional, right?
It is. That is correct.
And I have almost fully transitioned to using a espresso machine at home all the time. But my espresso machine can make an Americano, which I've talked about on the show before with Charlie. And so that's my version of drinking coffee now, because it's about the same volume as a cup of coffee. It's just made by pulling a shot of espresso and then filling it with hot water. When I have guests over at the house, I will still pull out the French press to make a French press. Or now that I have the espresso machine, I can make latte drinks and stuff, which I'm not very good at, but I try. And I use my French press mostly at this point to make cold brew for Julie, because Julie prefers cold brew.
Nice.
Yeah. And I still have a drip cone that I will do pour overs as well.
Yeah. Awesome. Well, Gary, we hope you enjoy your AeroPress and that it's been everything that you hope it is.
And thanks for writing in.
Yeah.
All right. So before we go, I have one very brief announcement. We are coming up one episode away from 300, and something we're going to try to do for episode 300. You will notice that when you go to the website, there'll be a little page that pops up before the main website loads that is requesting people to help us out with episode 300. We want to create a chorus of sort ofs. What that means is this was an idea that Karen had on Twitter and tweeted us about, and we thought, that's a great idea, Karen. We're going to do that. And so thanks to her for that. And what we want people to do is record themselves saying our classic, sort of sign-off line, and then we're going to put as many of those together into a giant sort of at the end of 300. Right. So... Like the page will be up online prompting people to do that. The method should be very simple, even for people that aren't that tech savvy. You can just record it on your phone and email it to us directly from your phone. Or if you want to record it on your computer, we're going to link you to a website that can transfer that file to us. So basically, if you could just get us a file with you saying sort of, please include your name so we have a rough idea of who is sending all these in, so we can just keep track of that for our own edification and enjoyment. But really, we just want to get as many people as we can recording themselves saying sort of and sending it into us. So the main email that you can use for that is PaleoPOWs@sciencesortof.com. That's PaleoPALS at SCIENCESORTOF.com.
I think you got it right.
Lost myself a little bit there, but yeah, it's the PaleoPOWs at ScienceSorter.com. It's also in the Contact Us page on the website, ScienceSorter.com. You can't attach a file to the Contact Us page, but if you use, we are recommending that people use a website called WeTransfer. So that is a website that allows you to upload files for free for anyone to download for 2 weeks. So you can get a link, and then you can use that to link us to the file for download. Or if you already use something like Google Drive or Dropbox and can send a file that way, that's totally great too. But the main thing is we just want to get as many as we can. We do still have the voicemail line active. That's 312-PaleoPOWs. That's 312-725-3672. So you can also just call in that number and leave a voicemail. The quality on that one's going to be a little lower, but we're hoping that if we get plenty of these, that will kind of all even out in the wash. So I just wanted to encourage everyone listening. If you listen to this right when it comes out, you'll still have about 2 weeks to get your sort of in. And I'm really anticipating having a wonderful blend of harmonies and voices and everyone who has enjoyed the show for 300 episodes or fewer.
Bonus if you get a friend or a stranger.
Yeah, or just do it at a bar or at your improv night, or
super bonus if it's at a bar.
Or like if go to church and get the chorus to actually sing it.
Triple bonus if it's the church choir.
Yeah. I guess we should maybe Abe and I will do something special for the person who sends in the most ambitious sort of. I don't know what it is yet, but we'll send you
absolutely
a beer or
will.
All right, cool. Well, with that, thanks for helping me out with this part of the show, Abe. I appreciate it.
Always.
And we'll see everyone for the momentous episode 300 coming at you soon, where maybe less so than usual, but I will do my best to promise that you will be able to get some science. Sort of. That was an example of how to do it.
Visit ScienceSortOf.com for show notes, links to all the stories we talked about, and ways to interact with the hosts, guests, and other listeners. Science Sort of is brought to you by the Brachiolobe Media Network of podcasts, with audio engineering by Tim Dobbs of the Encyclopedia Brunch podcast. That's all for this week. See you next time on Science Sort of.
So I struggle with the Scottish Micmac thing.
Yeah. So it's McAnulty.
McAnulty.
Yeah, you got it.
Okay. I did watch the video and you say it in the video, so that helped. But
that's good. Yeah.
It's been an issue on this podcast before. So
yeah, it's not phonetic.